Hi, my name is Dr. David Hudson, and today we'll be doing part one of a lecture on the introduction to marine science, and this is mainly focused for marine science for non-scientists. So to start, just want to frame out marine science as a discipline, and this is really rooted in humanity's ocean exploration over the course of millennia. So we're thinking about everything from you know, the curiosity and ingenuity of people who are mariners across cultures, across the world. And this is something that people brought with them and brought their curiosity with them. And this is not by any means exclusive to the European explorers like Cook and Darwin and the Challenger, but this goes back further to the exploration of new places for humanity to live and thrive. So just thinking in terms of ways where you're going across vast stretches of ocean, the Polynesian seafaring is one that is pretty incredible. They've gone across major distances with maps made of islands made out of sticks to guide them through these places with very few tools, but extremely accurate navigation. And as marine science grew with the tools, remember, science and technology follow one another, then further questions could be answered. So the growth in knowledge has been spurred by this ambition to find resources, to control those resources, and that extends to even now in our modern world. This went hand in hand with, and more recently, the development of marine laboratories. Some more modern ones would include the Marine Biological Laboratory in Woods Hole, where the first squid axon was recorded. The Stazione Zoologica in Naples in 1872, Marine Biological Society Laboratory in the UK, Hopkins Marine Station, which is run by Stanford University, Scripps Institute of Oceanography out in San Diego, and Woods Hole Oceanographic Institution, which started later, not to be confused with Marine Biological Laboratory. So these marine laboratories started to concentrate resources and collaborations so that one would be able to start to move across these different disciplines. So as we're thinking of what some of these new tools might be and what allowed us to start to ask some of these really deeper questions. One, comes to play with just the self-contained underwater breathing apparatus, or scuba, the aqualung being the most famous, and that goes back to the development during the war. The frogmen in the eastern part, or the western part of the Atlantic, and the development of sonar technology, which stands for sound navigation and ranging. And sonar essentially focuses as having a sending and receiving sound production mechanism produce a wave of sound to reflect across an object against objects on the ground. So what that does is it distorts the reflection, and one can create a picture of what's going on. So you're not going to run into a reef or run into uh, some unseen navigational hazard. Well, animals use this, and particularly whales, and on land, bats use this to help navigate in these spaces where one can't normally see. Now, thinking about the development of additional new tools, certainly research vessels and their capabilities to have a, a, a system where you have um, a set of standards like the UNOL standards, where you can lock into a very specific um, pattern on the deck of a ship and just have these modules that come on or off the ship. Certainly, the development of submarines and submersibles have allowed us to explore in the depths of the ocean. Saturation diving, like at Aquarius, and what's been proposed with Proteus. In the lower left, you've got the Aquarius Reef Base in Florida that allows one to to follow experiments and do multiple saturation dives over the course of a day, and do that for 30 days. You can observe things a lot differently when you're in the water the whole time rather than just 40 minutes to an hour and a half. Deployable sensors like I deployed this year is a light and temperature meter that we had on a reef in Colombia this year, and things like remote sensing. So you've got all these satellites orbiting the Earth and taking measurements of the Earth's surface. This allows you to remotely sense changes in algal cover, changes in forest cover on land, and productivity signals and temperature signals at their simplest. Additional to that, you have remotely operated vehicles, which allow us to go where our human bodies will not allow us to, and autonomous underwater vehicles, which can go for months on end, being autonomously deployed, and will use a certain pattern that's programmed in to be able to kick back information to the researchers. These allow us to really answer both questions that are on the scale of this individual coral reef or this individual station, or those that are basin-wide or worldwide. It's really quite incredible. Part of that system would be the Global Ocean Observing System, which has a lot of sensors deployed throughout the world. Everything from sensors that are deployed on ships and marine mammals to the types of things that are more stationary, like a buoy or like a deployable system. So we can put a lot of sensors on things now. They've gotten a lot smaller, and it's allowed us to really amplify what we're able to do with these tools. So to that end, we have all these tools, we have all this technology, but it's underwritten by our use of the scientific method. So essentially, we're using reasoning to describe, explain, and make predictions about the natural world around us. You've done this before. You had a flat you ran over something and you have a flat tire. You make the observation that you have a flat tire. It's fairly obvious. But then you think about, well, what could cause that flat tire? You could run an experiment, etc. But there are two pieces of reasoning, two types of reasoning that we use in the scientific method to make conclusions and make these predictions and explanations about the world around us. The first is, the induction, which are observational reasoning, and then deduction, which goes from making a general statement to seeing how closely that applies to the specific. So when we're thinking about the scientific method very generally, we make an observation of a problem and we form a question. We form a hypothesis, which can be both general and specific. The hypothesis is an explanation, a proposed explanation for that problem or that answer to that question. We usually will, in our process, make multiple hypotheses and test those. The important part of my hypothesis is that it's both specific and testable. And so we go through the testing and an experiment or a set of multiple observations. We go through and analyze those data from that experimental design and make a conclusion. And the hypothesis is either unsupported, which is effectively a new observation. We form a new hypothesis about our situation, or it's a supported hypothesis, and it allows us to further refine the testing that we're going for, and we're usually going to repeat those experiments to make sure that it wasn't just by chance that we ended up getting those results. Well, specifically, and I alluded to this on the previous slide, that we make multiple observations, we make multiple alternate hypotheses at the same time and go through these where we have these very general hypotheses that go through. And remember, we can have, with the flat tire example, multiple things that could cause a flat tire. So what this allows us to do is refine the causes of a particular phenomenon and further explain the natural world around us. So if we have these collections of hypotheses that are well supported, we start to put together a theory, and theory being the collection of hypotheses that we are going to go for and are going to use to explain a particular phenomenon. So we're testing a hypothesis. Let's think about coral bleaching. We've observed that corals are bleached. Uh, we're noticing that the corals around the world are turning white and dying. This is actually a picture that I took this year of an Acropora palmata in the Caribbean that is pretty well bleached. It's not 100% bleached, but it's pretty close. We've already think about a hypothesis that is a prediction statement that must be testable. So as we're thinking about this, if we're thinking about something like there are mermaids in the ocean, well, we can't test that. It's very much a prediction statement, but it is not a testable prediction statement. So in this case, we're going to make an observation that the corals around the world are turning white and dying, and multiple hypotheses. So corals are dying because the climate is changing. Okay. Corals are dying because water conditions are bad and they like clean water. All right, that's possible as well. And corals are dying because water temperatures are outside their survivable limits. So these are all hypotheses, but there are various levels of being able to test these. So when we're thinking about testing hypothesis of coral bleaching, let's think about what a healthy coral looks like. And this is a healthy Acropora palmata from just around the corner from this other one. It has coral polyps that have zooxanthellae. These are essentially an internal garden. They live in very nutrient poor seas. So these are animals that need to feed. And if they don't have food, they will die. So instead of being able to feed, they actually have this garden inside their tissues that allows them to get nutrition, which is why Caribbean corals and tropical corals typically are hermatypic corals. They need to have these zooxanthellae. And if they get stressed, if they get perhaps sick, they may expel those zooxanthellae. And that is what a bleached coral is. This coral is still alive, but it no longer has its zooxanthellae, which means it is currently starving. So it can go back to being a healthy version of the coral if it takes up zooxanthellae from the environment, which newly settled corals do all the time, and bleached corals do as well. Or if they remain in this bleached state, they will continue to devolve and utilize the stores that they have, and basically get out competed by turfing algae. The coral polyps die, and then algae covers the skeleton. So it's and that is not a reversible scenario. So other tools that we can use to test this hypothesis of coral bleaching are things like remote sensing. So NOAA has a probability model for coral bleaching and heat stress that goes out once a week. And they have both maps for a 60% probability of bleaching and a 90% probability of immediate bleaching. This is directly related to sea surface temperatures. So this is an example of using the remote sensing technologies that we have to make some predictions on where we should deploy resources for effectively responding to coral bleaching events. You'll notice that I've been locally in northern Colombia of late. These are areas that haven't had as much of an issue with coral bleaching for this reason. They're certainly on a watch, and we've experienced it a lot, but the bleaching these past few weeks has been in some of the other areas. So essentially, the conclusions that we then make after these tools are used for an experiment or for really just repeated observations, we end up with the results of lots and lots of observations, and we may find that multiple alternative hypotheses are at work at the same time. So, in the sense of this particular coral meadow in Colombia, we may find that bleaching may have multiple causes, and that it may have some instances where temperature comes along, but we may also see other types of bleaching when there are rain events and other types of issues. So the importance for this is that we want to make sure that we aren't sticking with a very broad type of hypothesis like corals are dying because the climate is changing, because it's not really something that's truly testable. Even water conditions are bad. It's like, well, what about the water conditions are bad? How do we define that? We need to get it down to specific one or two factors that would be something that we can test. So what we would, if we would take a look at another example and think about the example that is often given as a classical instance here, we look at oyster growth and survival at two different sites. We take oysters from different sites. We have a warm site and we have a cold site, and we look at all these different types of variables that could be affecting growth and survival. You've got oyster type, so we want to make sure we're just focusing on one species. We've got temperature, we've got food, we've got salinity, we've got water quality, diseases that could be present like dermo or MSX, and other completely unknown factors that might be present in these locations. So when it comes to how we can think about growth and survival, oysters from different sites could perform differently. So we want to see whether temperature is the thing that allows us to do that. We could also look at food. But the idea here is that we have lots of variables that we would want to hold constant. We want to control all of these variables and test just one thing, just one factor. And at tops, if you want to really make a lot of work for yourself, test two factors. But then, when it comes down to being able to really answer a question whether this is something that we would want to do and whether it would be something that would be able to be tested, this is one of the examples of what we would have. So in a field experiment, we might actually do this with oyster type by controlling for reciprocal transplant. So we would take some oysters from the warm site, plant them at the cold site, and some oysters from the cold site and plant them on the warm site, or grow them in a common garden of cold temperature and a common garden of warm temperature, completely ex situ to the field. So we would do that in a controlled scenario. So we really know whether there's a difference in those two populations. So essentially, we're trying to get the same. We've got the same size oysters. We're trying to figure out whether there is this effect. So at the end of all of this, we end up with a collection of evidence that builds into a scientific theory. So the scientific theory allows us to explain the world around us. These are supported by overwhelming evidence, and they provide this really comprehensive explanation of how the world works. They are groups of supported hypotheses. These are, very refined ideas of how things work. It's the model that we have for how the world works around us. Some examples would be that plate tectonics and continental drift are a scientific theory. Evolution, gravity. Look this drops, right? So that's technically a theory, but at one point we had to do multiple observations to then essentially build this theory out to explain the world around us. Heliocentrism, the fact that the sun is at the center of the solar system, was a theory that had to be supported with multiple observations. But in science, there are very few absolutes because we're explaining a really messy world. And in any model, we want to make sure that we have the least number of explanatory factors in that model to make sense of it all. And it does have its limits. Science cannot offer input on values, feelings, beliefs. It's just outside of science. It's not something that can be explained under science. So with that ends part 1. I will continue up with the next part at your leisure. Thanks for watching. If you'd like to support us, please go to buy coffee at www.upwellcoffee.com or go to remoteecologist.org/support to donate to our nonprofit or to join us in the field on trips. Thanks so much.